present you Professor Jesus Simal. Professor Jesus Simal is a full professor in food science at the University of Vigo. He is also the leader of a research group of excellence at Galicia. He is also the leader of Environmental, Agricultural and Food Research Center formed by 10 research groups from different fields. And he is also the head of the Department of Analytical Chemistry and Food Science at the University of Vigo. So, Professor Jesus Simal, you have 25 minutes, more or less, to perform your presentation. So I will pass you the word to hear your interesting keynote.
Yes, can you see my presentation?
There we go. That's it.
Okay, thank you very much. I want to thank to the organizers because I'm very happy to be here, even online, and be part of this interesting Congress. My talk is about metabolomics, the basics and applications in food and function, and also medical applications. We intend to define what is a metabolome, to see the importance of potential application of metabolomics, to talk about the operational principles of the main technologies, and also to understand the difference between targeted and untargeted metabolomics. If we see the pyramid of life, in the base we have the genome, then in the upper part proteome, and in the more upper part metabolome, and the influence of physiological and environmental factors are higher in the upper part of the pyramid. What is a metabolome? Genomics is the field of life science that use high throughput technologies to identify all the genes in a given cell, tissue, or organism. That is the genome. In that way, metabolomics is the field of life science that use high throughput technologies to identify all the small molecules of metabolites in a given cell, tissue, or organism. That is the metabolome. But why is metabolomics so important? Because metabolite, it can be any organic molecule that is detectable, usually above picomolar in the body, with a molecular weight lower than 1,500 Daltons. Includes peptides, oligonucleotides, sugars, nucleosides, organic acids, ketones, aldehydes, and along, etc. Includes both human and microbial products. It should be detectable. Can be detectable above 1 femtomolar, 1 picomolar, 1 nanomolar, depending on the technique. Then what is a metabolome? The complete collection of a small molecule metabolites in a cell, organ, tissue, or organism, but includes both endogenous and exogenous molecules, as well also as transient or even theoretical molecules. The molecules that can be detected are very dependent on the detection technology. That is the reason we say that the metabolome size is always ill-defined, very poorly defined. If we look at the pyramid of life, we see that many food-related products have different metabolites. In an order of magnitude, for example, plants are already well known, 200,000 chemicals. For microbes, 60,000 chemicals around. And for mammals, at least 20,000 chemicals. There are a lot of databases for metabolomes. There are databases for toxins, environmental chemicals, for drug metabolites, for drugs, for food additives, for phytochemicals, for also endogenous metabolites in the range of femtomolar to molar. But there are also theoretical human metabolomes, like those formed by lipid and lipid derivatives, also those formed by drugs, from food components, food ingredients, and also secondary endogenous metabolites. Why is metabolomics important? Because small molecules count. More than 95% of all diagnostic clinical assays tests for small molecules. 89% of all known drugs are small molecules. 50% of all drugs are derived from persisting metabolites. 30% of identified genetic disorders involve diseases of small molecule metabolism. Small molecules serve also as cofactors and signaling molecules for thousands of proteins that also are with enzymatic or metabolic activities. We say that metabolites are the canaries of the genome. You know, this bird, the canary, is a very sensitive bird. In this way, we say the metabolites are the canaries of the genome because a single base change in the DNA can lead to 10,000 changes in metabolite levels. For example, the response in time of the genome can take days, weeks. The response in time of the proteome can take days, hours, but the response in time of metabolomics, of metabolites, can be in just seconds, minutes. It's very fast, the response. The key question is that it's not so easy to understand the whole thing of metabolic pathways, interconnection of metabolites. It's a really complex picture. But the interconnection between genome, proteome, and metabolome can help us in this process of understanding metabolomics. The metabolome is connected to all other omes because small molecules like adenosine monophosphate, cytosine monophosphate, guanosine monophosphate, etc., are the primary constituents of the genome and transcriptome. Small molecules, like the 20 amino acids, are also the primary constituents of the proteome. Small molecules, like lipids, give to the cells their shape, form, integrity, and structure. Small molecules, sugars, lipids, amino acids, adenosine triphosphate, are the source of all cellular energy. Small molecules, too, serve as cofactors and signaling molecules for both the proteome and the genome. That is why we say that the genome and proteome largely evolved to catalyze the chemistry of all small molecules. The interplay connection between genomics, proteomics, and metabolomics with the help of technology, information, and communication tools like bioinformatics or cheminformatics can help us to understand the system biology. The metabolomics workflow is that way. We start with a biological tissue, a food sample, we do extraction, we analyze the extracts with different techniques, and then we need to do biological interpretation of all data obtained. But why metabolomics is so difficult? Because genome is just a matter of 4 bases in the base of the pyramid. Proteome is proteins are constructed based on 20 different amino acids, but metabolites are a large quantity of molecules, 200,000 molecules. The chemical diversity can increase from the lower part of the pyramid to the upper part of the pyramid. We have a lot of different techniques to understand metabolomics. We have ultra performance liquid chromatography, capillary electrophoresis, microfluidics, different combinations with mass spectrometer detectors, nuclear magnetic resonance spectroscopy, X-ray crystallography, laser-induced fluorescence detection, and etc. For example, in chromatography, the basis is we have an stationary phase, a mobile phase, a column, we inject the sample, we have a partition between both phases, and we have a retention time for all the components, and with appropriate standards, we can identify the molecules in a certain sample, and measuring the area of the signal, we can also relate to the quantity of the molecule. If we use mass spectrometer detectors, at least the molecular weight of the different components is very characteristic for these components. But when we use a mass spectrometer detector, we don't use the average mass, that is the weighted mass of all isotopic components, like the one in red. We use the monoisotopic mass, the mass determined using the masses of the most abundant isotopes. And usually we have an isotopic distribution. To see this isotopic distribution, we need high resolution instruments. For example, a time of flight mass spectrometer is 10 times higher resolution than ion trap mass spectrometer. Here in the ion trap, we cannot detect the isotopic distribution, but with the time of flight, it is possible to see that isotopic distribution. In electronic path, when you use a gas chromatograph with a mass spectrometer detector, for example, if we want to detect the spectrum of methanol, we bombard the methanol with electrons. We take one electron from the molecule, we have a molecular ion, and this is fragmented into the most stable cations. We have a unique fingerprint for this component. It's like a digital track for that molecule. That is also very characteristic. In NMR spectroscopy, nuclear magnetic resonance measure nuclear magnetism, or better to say, change in nuclear magnetisms in a molecule. NMR measures the absorption of light, radio waves, due to changes in nuclear spin orientation. NMR only occurs when a sample is in a strong magnetic field, and different nuclei absorb at different energies, frequencies. For example, we need a magnet to expose our sample to the magnet, and increasing magnetic field strength, we have also higher sensitivity to detect lower amounts of the different components. If this is a typical NMR spectrum, we have trimethylsiloxane in a proton NMR at a reference material with 0 chemical shift. And then we have different chemical shifts for the different functional groups at different frequencies. We have also a splitting pattern produced from the spin coupling that is due to the environment of the functional groups within the structure of the molecule. And we have also different peak intensities depending on the amount of functional groups in the molecule. For example, we have trimethylsiloxane with 0 chemical shift and different functional groups with traditional frequencies that are characteristics for them. In this case, then we can interpret a spectrum like that one from ethylbenzene. This is the signal for benzene, for ethane, for methyl groups, etc. All these technologies has different advantages on robots. For example, NMR is not very sensitive in comparison to the other. Its sensitivity is in the range of micromolar to molar. Then it's very suitable when we try to measure non-components. But if we want to identify unknown components, then we should record to LCMS, direct injection MS, because the sensitivity is in the range of femtomolar, picomolar, nanomolar. Also, the different techniques has different advantages and drawbacks. For example, NMR is very suitable for water-soluble molecules. Instead of it, direct injection mass spectrometry is mainly useful for mainly hydrophobic molecules. Also, the sample volume we need is very different from one technique to the other. If we also compare sample preparation time, runtime, data analysis, limit of detection, and number of metabolites that can be detected, direct injection mass spectrometry is a very interesting technique. We have two ways of approaching metabolomics. We have target methods. We know already what we want to measure, and then we do analysis to identify with security those components and quantify those components. These are target methods, quantitative methods. But when we do not know what is going on, we look for untargeted methods, profiling methods, chemometric methods. The approach is different. For example, in profiling and target methods, we do sample preparation, extraction, we do data collection, and then we use chemometric procedures to see difference, to see what is important in the treatments against control, and to monitor our interest in the differences, and then to look at these molecules and try to identify them. In quantitative target methods, we start also with sample preparation, but we already know what we want to measure, what we want to identify and quantify. We do identification and quantification, and then we do data reduction to understand the connections between all these metabolites to be able to do biological interpretation through metabolic pathways. What we try is to move from data, spectra or chromatograms to lists of components that are very clear, identified. And from the list of components, we want to move to pathways to do the biological interpretation that is very complex. And from the list of components and the metabolic pathways, we use models to detect biomarkers for specific treatments or specific health states. What is key in every step is the following. When we move from data, spectra, chromatograms to leads, we have to be very sure of the integrity of our data, the quality of our data. We have to remove noise. We have to classify properly the data, to normalize the data, to assess the real significance of the data, and be sure of certain metabolite identification and also quantification. When we try to move from lists of components to pathways and to detect biomarkers, we have to be very sure of the mapping in the pathway, to be sure of the biological interpretation. We have lots of possibilities for metabolomics. All of these in the screen are possible: food and beverage tests, nutraceutical analysis, drug phenotyping. I was just selecting 4 examples in red. And I want to pose you a few examples just to see how it works. For example, medical metabolomics is a very important field today because it's possible to generate metabolic signatures of disease states or host responses, for example, to a drug or a bioactive ingredient. It's possible to obtain an holistic view of metabolism and the effects of a specific treatment on metabolism. It's also possible to accelerate assessment and diagnosis of a functional problem or a disease. It's a very accurate, cheap, rapid form of identifying disease phenotypes or the response of a host, of a patient, or people to bioactive ingredients of the drugs. It's also possible to monitor gene-environment interactions. And it's also possible to track the effects of a drug, a bioactive ingredient, or surgery. For example, in the field of metabolomics and drug toxicology, we have here an example of the different metabolic response of a group of people, that is the control group, and people exposed to different analgesic drugs. Then we have a different trace of responses that can be easily detected, the components that can be different between treatments by chemometric techniques like a principal component analysis. Also, we can use functional magnetic resonance to identify in a certain organ, for example, the brain, what kind of metabolites are produced, or even the effect of certain bioactive ingredients or metabolites that we enter into the body in the different regions of the organ, in the different regions of the brain. We can also do metabolic microarrays. For example, we can expose different people, different patients to different treatments, and we see the different response, the metabolic response of the different people. In the case of food, we can give to the people, for example, strawberries with polyphenols, and we can measure the metabolites in the gut. Then we can have metabotypes for the different people because of the different microbiota in the different people. Metabolomics is also an important field for transplants because we can use non-invasive samples like urine, blood, bile. It can be very quite organ specific. The response is very fast, 60 seconds, just seconds, minutes, not like gene, protein, and tissue changes that happen in minutes, hours, or days. It can also be simple to monitor patient or organ function before and after the operation. For example, in the case of chronic renal failure, there are different biomarkers. And we have lots of biomarkers for kidney failure. These biomarkers can be related to different states of the failure. For example, creatinine is related to late stage organ stress. Trimethylamino oxidase, it can be related to early stage responses. Other metabolites can be related to tissue damage, others to oxidative stress, and some others to immune response, inflammation, etc. There is a large metabolome project running on. It was started in Canada, but now it's an international project It got started in 2005. The mandate of this huge project is to quantify both normal and abnormal ranges of metabolites in all kind of fluids, body fluids like urine, cerebrospinal fluid, plasma, white blood cells. That has to be freely available, electronic available for all public. The purpose of all this is to facilitate metabolomics, to improve identification of a disease or the health status of a people, the prognosis, that is to say how it's going to evolve the disease, the prediction, to do disease monitoring, to test for new drugs or new bioactive ingredients. It's also important for the linkage between metabolome, genome, proteome, to understand system biology, and also to develop tools like software tools to understand what is so complex, metabolomic pathways. To conclude, I want to give you some clear statements. Metabolomics is increasing, rapidly becoming the new clinical chemistry, complements genomics, proteomics, histology, and is so quick producing change that is easy to detect many changes after exposure to a chemical contaminant, bioactive ingredient, drug, or whatever. And that's all. That's all. If you have any questions, please do. I'm here to be useful if it's possible. Thank you.
Thank you very much, Professor Simal, for your wonderful presentation. I have here just a questions for you. Dr. Carosho is asking, what is your opinion about using metabolomics as a tool for authentication and a measure of quality control of the production processes? Talking, of course, about the food sector.
Yeah, metabolomics is also very important in food control, above all in the area of authenticity, food fraud, to detect fraudulent food products. And there are two ways of approaching this, like I showed you before. Targeted metabolomics, to go to monitor something that is clear, a biomarker of a certain process or food product, or food fingerprinting, to use raw data, just chromatograms, spectra, and to do variant analysis of chromatograms, spectra, to detect fingerprintings that are clear biomarkers of the authenticity of a product, or of a product that is a fraud. That are the most useful approaches.
Thank you, Professor. Thank you, Professor Simal, for your answer. So